Hi everyone! This video is to walk you through the central Dogma Lab. So at this point, we've started our molecular biology cycle of study, and we've mostly been focusing on DNA. When we think about the central dogma, we need to remember that DNA codes for RNA, and then RNA is translated into a protein. So DNA stores the genetic information, and of course, when we turn it into a protein through RNA, those proteins are then the functional units of the cell. They're really the ones doing all of the work. So in that way, proteins help us express our genes. They help us see what traits we have, how we behave, what we look like, what types of diseases we might be predisposed to. So the genetic code is our DNA, but through the central dogma, we translate it into RNA and then turn it into proteins. Those proteins then go out and do all of the functional work. So as you can imagine, your set of DNA in your cells is incredibly important. That's what tells you what you're going to look like, what you're going to do, right? We pass that DNA along to our offspring in a certain form. Okay, of course, it combines with whoever your partner is a little bit, but every living thing has DNA, whether it's a bacteria cell, a fungus, whether it's a tree or a human being. That DNA is all about the sequence of the nucleotides, right? Remember, DNA and RNA are nucleic acids, and they are made up of the monomer nucleotide. A nucleotide, of course, has three parts. It has the phosphate group, the 5-carbon pentose sugar, and then the nitrogenous base. And so your 5-carbon sugar and your pentose group are what make up the backbone of the DNA molecules. We call that the sugar phosphate backbone, and that binds together to make the structural outlines of both strands of DNA. Now, we say that DNA is double-stranded, and it's usually represented as a ribbon or a line in pictures. So when you see that line in a picture of DNA, that's going to be your sugar and phosphate backbone, the sugar and phosphate from your nucleotides bonded together. Now, what about the nitrogenous base? So at this point, you know the nitrogenous bases can be one of 4 different things: adenine, cytosine, guanine, and thymine. And whichever nitrogenous base is attached to that nucleotide is going to determine which one it bonds to. Okay. Of course, our nitrogenous bases are the ones that are pairing up with their complementary pair. So A and T are complementary. They're always going to match up. And C and G are complementary. They are always going to match up, right? So this is for DNA. That's how we know what the 2 strands of DNA are. They're not identical to each other. They complement one another. So instead of mirroring each other, having the exact same nitrogenous bases for every single line, they're instead going to be matched up with their partner, their best friend. So we're going to take a look at that today. This really isn't so much an experiment, but much more of a worksheet to help us review. So take a look at your procedure. Make sure you read through it all before you get started. And again, this is for the Central Dogma Lab. Now, it starts out with a really nice review of tonicity, and of course, how different concentrations can affect movement within cells. So part A, it says a review of the solutions and membranes. Review your notes, use your textbook if you need to, and as always, come to recitation with questions, make tutoring appointments with questions, and email me with any questions you might have. Now, number 1 says, diffusion is when a substance moves from areas of blank concentration to blank concentration. My cat's joining us today. Excuse me, kitty. So what is diffusion? Is it going from low to high or high to low? Give me just a minute. Okay. Think about it in your head. Is it active? Is it passive? Do you need energy or not? Now, your next question is asking about the diffusion of water. We have a very specific name for that. If you're forgetting some of these concepts, this was our transportation section in our cellular biology cycle of study. So you can always go back and read through that again. These concepts are going to come up again on your competency exam, so they're really important to remember and to be reviewing regularly. So hopefully this will help you a little bit. Now scrolling down, we have some really nice pictures of red blood cells in a beaker, okay, obviously not to scale, but these are going to be different types of tonicity. And remember, tonicity is all about concentration. It has to do with how many particles are in the solution. And as a reminder, when we talk about tonicity, we're talking about the concentration of the solution, not the concentration inside the cell. That's really going to help you change your answers to get the right one. So looking at these pictures, you want to label all three of those beakers. So beaker A, you can see the arrows look relatively equal going in and out. Look at the shape of the cell, the size of the cell. What is the tonicity of A? Looking at B, we see a much bigger area going into the cell. We see a really big cell. What is the tonicity of B? Remember, thinking about the solution, the concentration of the solution, not the concentration of the cell. And then in C, we see a whole bunch of blue dots. We see kind of a shriveled up cell, and we see a big arrow going out. So what is the tonicity for that? Make sure you write down those three names in the boxes below the beakers. And then it says which solution is likely to lead to hemolysis, or the breaking apart of a red blood cell. So of those three beakers, what's the tonicity of the one that could lead to a lysis? Number 2, you want to put some definitions down. Make sure that your definitions are thorough and not copied straight from your book or the internet. If you copy something verbatim, meaning word for word, that's of course going to be plagiarism. So start practicing putting things into your own words, thinking through the concepts and digesting them. When you give me these three definitions, make sure that they are complete. They're defining the entirety of the word. And number 3, describe the role of the cellular membrane. So at this point, you guys have been college students for a little while. We're nearing the end of our 6 credit hour biology science class. So you should be able to give me complete sentences, free of typos, and you should be able to give me a complete idea of what the cell membrane does. So when you go through this lab and you start writing out different things, remember this is a great study tool for you, so be thorough. You just need a few sentences here if you're getting them as succinct and fulfilling as you can. So what is the role of the cell membrane? What are all the functions that we talked about? Why is it important? What does it do? Again, complete sentences, and make sure you check your spelling as best you can. That's just a way to be professional as we move into our college classes and later into our careers. Now, number 4, it says, in regards to the surface area, which is of course the cellular membrane, and the volume, which is all the stuff inside the cell, explain why it's important for most cells to remain small. This all has to do with your outside the cell lab that you guys did. So you had multiple experiments where you got to figure out why cells stay small, what happens if they're too big. Using that knowledge, write a few sentences on why cells need to stay small. Hint, what is the role of a cell membrane? Can it do its job if it's too large? If the cell is too large? Okay, why or why not? So more than just a couple words, give me a few sentences at least. Thinking back to the outside the cell lab, what does the surface area to volume ratio have to do with the cell? And then we're going to look at part B. So part B is talking about a type of disease called sickle cell anemia. Probably some of you know about this. Maybe you have family members or friends who suffer from sickle cell anemia. It certainly is more common than we would like, of course, and it's a disease that can be quite impactful on a person's life. So it can really affect what type of activities you're able to do and even the lifespan that you have. Now, if you've never heard of sickle cell anemia before, we're going to talk about it a little bit and you'll have some time to research it. So this is a good point to get on your computer, look up some reputable sources, .edu, .org type sources, and start thinking about what exactly this means. So when we think about the sickle shape, maybe you think of a sickle moon. It's that curved shape, right? Not a complete circle, but just that curved kind of C shape. Our red blood cells normally would be nice and round and then kind of flattened down like a donut without the hole taken out, okay? But they're going to be in a circular shape. Sickle cells have lost that circular shape. They're much more like the C shape. Okay. Now think about what red blood cells do for the body. They carry oxygen through the bloodstream, and they need to have a lot of room for oxygen to bind to the hemoglobin inside the red blood cell. So if we have a different shape of a red blood cell, it's going to affect how much oxygen we can carry and also how much carbon dioxide we can carry to release as waste. So keep that in mind as we read about it. Sickle cell anemia is just a disease that we chose because it's based off of a mutation in the DNA. So thinking all about how our genetic code can then affect how we function and what types of diseases we might get throughout our lives. Okay. Sickle cell anemia is much more prevalent in certain communities. African American population tends to have a rather higher presence of it in their genetic code. Some people can be carriers for sickle cell anemia, but not actually expressing the disease. So they might have a little change in their DNA, but not enough to make sickle cell shapes. And then some people are not carriers for it at all. So as you do your research, maybe you'll come across different things like that. Here in Cincinnati, we actually do have a pretty big sickle cell anemia research center. And so they're working on how to fix this, how to make people's lives a little bit more manageable. Living with this disease can be quite painful. My friend's sister has it, and she's in and out of the hospital a lot. She's only 14 years old, and her family's very worried about what her future life is going to look like. So again, maybe you know someone with it. If you don't, take some time to research it and really understand what this disease is, because chances are you will see it at some point in your healthcare careers. Now, part B of your lab says, why does everyone make such a fuss over DNA? What's the big deal? DNA is a polymer made up of nucleotides. It stores our genetic material, and it tells the body what types of proteins to make. The proteins are polymers of amino acids, right? They're made up of about 20 to 21 different types of amino acids. And it's all about the order of the amino acids that determines what protein it will make. So if we put the amino acids in a different order, we could get a different type of protein. It's similar with DNA. If we put our nucleotides in a different order, we're then going to make different types of proteins because the DNA codes for the RNA, which codes for proteins. So if we have an error in the DNA, that could be carried down throughout the central dogma. And then our proteins, those functional units of the cells, could end up having issues. Now it says, if you were to be dehydrated, much of the fibrous material left in the body is protein. So if we ignore all the water, we have a lot of protein in the body. And who told your body which proteins to make? Well, it was the DNA, right? The DNA is the blueprint for the cell. It's the instruction sheet telling the body what to do. So in a way, DNA essentially makes us who we are, right? It tells us what we look like to an extent. To an extent, it tells us how we're going to act. Now, a lot of that goes back to the whole discussion of nature versus nurture, if you've heard that phrase before. Nature being how much is in our DNA, nurture being how much is in our environment, right? We can certainly change things about our personalities, about our health, about our predisposition to diseases, just by the actions that we take and the thoughts that we have. So even if our DNA says one thing, it doesn't always mean that that's the rule. We do have some control and flexibility around it. But in a way, DNA is coding for all of the proteins being made in the body. And so it's an incredibly important way to tell us what will happen throughout our bodies. Now back to sickle cell anemia. It says hemoglobin is a type of protein, and it makes up about a third of your red blood cell. So it's a pretty important protein in our red blood cells. It helps those red blood cells carry oxygen. So we breathe oxygen in, and we need it in our tissues in order to make ATP, in order to make energy. So our red blood cells have the very important job of bringing oxygen to our cells so that we can make energy. We then have to expel our carbon dioxide, and so the bloodstream helps us do that too. If we get too much carbon dioxide, a waste product, building up in our body, we're going to get very sick and eventually could die. So hemoglobin accounts for about a third of a healthy, normal-sized red blood cell, and it's carrying a lot of important molecules back and forth in the body. So our assignment today is to compare how the normal allele for hemoglobin is expressed and how the sickle cell allele for hemoglobin is expressed. So if the word allele is new to you, an allele is a type of gene. So we have this whole big strand of DNA, this big genetic code. We call that our genome, right? The human genome. One section of that, the section that codes for one particular protein, is called an allele. So an allele is a section of DNA that codes for a protein, and if we have different variations from person to person, it's an allele. So I might have an allele for brown hair, you might have an allele for red hair or for blonde hair. So any type of section of DNA that's coding for a protein, a gene we could call it, if there are variations from person to person, we call it an allele. It's a section of the DNA, not the entire thing, and it could vary from person to person. So I have the normal healthy allele for full-size red blood cells, as far as I know. But maybe someone else has the sickle cell allele, so the same section of our DNA that codes for how our red blood cells are shaped. One person's could be a normal shape and one person's could be a sickle cell shape. That difference is called an allele. So it says all of the steps from the DNA to the protein's functioning is going to be explored in the lab. We will determine what the difference is between the sickle cell allele and the normal allele. So we're going to look at the DNA and see what the actual difference is from one to another. And of course, your definitions here. So a gene is that segment of DNA that codes for a protein. So say we have our 2 meters long of DNA. Maybe just 1,000 nucleotides of that. So just a small chunk of it codes for a specific protein. Let's say it codes for your hemoglobin protein. That's your gene. But we can have variations in that. That section, that gene, could look different from person to person. And so that's when we call it an allele. When it starts looking different, it's a form of the gene. It could make a certain protein, or it could make a slightly different type of protein. Okay, still for the same functioning part of the body. So down below in bold, we see DNA for a normal allele, right? This is all about red blood cells, okay? Now this is a very, very small portion of the DNA. We just took out the little section that we want to look at. Remember, DNA is a very large macromolecule. But DNA for a normal allele has this specific coding, okay? So we see our codons. Codons, of course, are the 3 nucleotides in each little section. So CAA is one codon, GTA is the next codon, and all the way down. And then next to it, we see the DNA for the sickle cell allele. So these are coming from the same section of DNA, but in 2 different people. And so they're 2 different alleles. One might be slightly different than the other, as we're going to look. Take a look at these 2 different segments of DNA. Do you notice any differences? Is anything different between those 2 lines? Now looking at number 1, it says use the following gene sequencing tables to determine the proper sequence for the mRNA and write that sequence in the appropriate box for the normal allele and for the sickle cell allele. Okay, what does that mean? Well, we don't need any type of sequencing table because when we go from DNA to messenger RNA or mRNA, we know what it's going to be just by looking at the patterns. Okay, so think back to our lecture where we sequenced DNA. We took one existing strand of DNA and we replicated it by making a complementary strand during the synthesis phase of interphase, right? So we were given a strand of DNA and we matched it up with its complementary base pairs so that we ended up having our 2 strands of DNA, a nice, happy, complete molecule. That's for synthesis. That is for replicating our DNA, going from one DNA molecule to a second DNA molecule so that we can divide the cell and make more cells. What we're going to be doing right now is slightly different. We want to go from DNA into messenger RNA, okay? Then we're going to go from messenger RNA into the amino acid. So we are going to be doing transcription, DNA to messenger RNA, and translation, messenger RNA to amino acids. Transcription and translation, right? Now, remember, RNA has a different type of amino acid base pair, a different type of nucleotide base pair. RNA has uracil instead of thymine. So if you have an RNA molecule, you should not have the letter T anywhere in that sequence. Anywhere that a T would exist, you're instead going to put a U for uracil, okay? So I have a strand of DNA for my normal allele. In your table, you're going to copy what's given to you up above. So my normal allele is given to me on the left side in bold. I'm going to copy that down into codons. So in my very first box of this table, I'm going to write CAA. My next box, I'm going to write GTA. The next box, I'll write AAC, and all the way down. You want to have all of your codons written down in that table. Once you do that, you're then going to transcribe it into messenger RNA. So your second line of the table where it says mRNA, you want to fill in those boxes with the complementary base pairs, but keep in mind you should not have thymine at all. So CAA, what will be the matching codon? What will be the complementary codon for messenger RNA? If DNA has a C for cytosine, what will messenger RNA have? Well, C always matches with G, guanine. So write a G, okay? Your C and your G match together. Now CAA, what about our A's? A's typically match with T, thymine, but remember, we don't have thymine in RNA. Instead, we have uracil. So anywhere you see an A for the DNA, you're going to match it with uracil for your RNA. So in your first box, you should have CAA for DNA, and its complementary codon for messenger RNA is GUU. Okay? Now let's do the next box together. GTA is my DNA codon. My matching complementary messenger RNA codon, GTA was DNA. G is always going to match with C, so I'm going to write a C. T is always going to match with A. That stays the same. And A is allowed to exist in RNA, so I'm going to write an A for my next letter. GTA, my last one is A. Well, A matches with thymine or with uracil for RNA, so I'm going to write a U. So to repeat, my second codon for messenger RNA is CAU. Okay? Try that with the rest. Now, once you've done that, you're going to need something called a genetic code table. My cat is making my legs fall asleep. Okay. Now that you've made your DNA and your RNA lines, you're going to need your genetic code table. The genetic code table is something that you can just Google. You also have one in your book, but you can just go on the internet and search genetic code table, and you can use that, right? It's very common. It exists for any time we need to translate DNA. So pull up your genetic code table and use that to help you write out the amino acid sequence. Now to do this, you're going to look at the messenger RNA codon. Okay, we're done with DNA at this point. We've now made our messenger RNA. The messenger RNA is going to leave the cell. The messenger RNA is going to leave the nucleus. And once it leaves the nucleus, it's going to go to a ribosome. At the ribosome, we start to actually make our proteins. Kitty. All right. So your messenger RNA has left the nucleus, and we're going to start our translation, going from messenger RNA to an amino acid. Using your genetic code table, you're going to take the codon of messenger RNA, and you're going to see what amino acid it's coding for. So if our very first codon of messenger RNA is GUU, guanine, uracil, uracil, look at your genetic code table. Your first base pair is going to be guanine, so find guanine. then match it up with the second one of uracil and the third one of uracil. When you see GUU, it's going to give you the amino acid that it will code for. Okay, does everyone see that? This is going to be valine. So in your table, underneath GUU, you're going to write valine, or VAL. You can use the 3-letter abbreviations here if you'd like. Okay? You're going to do that for this entire sequence. So my second messenger RNA codon is CAU. Look for CAU on your genetic code table and write down what the amino acid should be. Once you have that table complete, you're going to do the exact same thing for your sickle cell allele. So looking back up above, we have the DNA written out in bold for the sickle cell allele. You're going to copy that down into the box below in the DNA slot. So I'm looking at the sickle cell allele table. On the DNA line, I'm going to write CAA. That's my first codon. Then I'm going to write GTA. That's my second codon. All the way down until I end with CTT in my last box. Now you'll notice you're going to have a little bit of overlap between this table and the one that you just completed. So take a look at your DNA codon, transcribe it into messenger RNA. Put the complementary pairs in there just like you did above. Remember, uracil should be there instead of thymine, but that's the only change. Complementary pairs, so match them up with their partner, and then use your genetic code table to translate from messenger RNA into the amino acid. Okay. You now have a completed table for your normal allele and for your sickle cell allele. You should have all of those boxes filled out, all of your DNA, mRNA, and your amino acids. We have finished the translation, so we've now made our polypeptide chain, which is our strand of amino acids. This is what turns into our protein, right? So number 4, we're going to take a look at these two tables and compare. How many differences are there between the normal allele and the sickle cell allele? So for DNA, take a look. How many differences are there? You see 1 difference, 2 differences, 5 differences, 10 differences. How many differences? What about the messenger RNA? Compare the messenger RNA for the normal allele to the messenger RNA for the sickle cell allele. How many differences? How many letters are different? Write down the number. And then looking at the amino acid sequence, when we look at the amino acid sequence for the normal allele versus the amino acid sequence for the sickle cell allele. How many amino acids are different? Are any of them? Is 1 different? Are 5 different? Write down the number. Now number 5, it says since hemoglobin makes up a large part of the red blood cell, having a problem with the hemoglobin can greatly affect our oxygen transport. The single amino acid difference in the hemoglobin changes the structure of the entire protein, right? The sequence of amino acids determines the shape that the protein will take, which determines the function it can have. So, the difference in the amino acid from the normal to the sickle cell allele changes the structure of the entire protein, which then changes the structure of the entire red blood cell, especially under low oxygen conditions when we don't have oxygen binding. So the sickle cell hemoglobin clumps together when we have a low oxygen condition, and it kind of scrunches up the whole red blood cell, hence giving us the name sickle cell anemia. So do some quick research, look up what these shapes actually look like, and draw them below. Draw a normal red blood cell, draw a sickle-celled red blood cell. Okay, I'm on to number 6. It says mutations happen often during the replication of the DNA molecule. Okay. There are checks that catch many of the mutations, right? We have DNA polymerase who proofreads it. We have checkpoints after the synthesis phase. However, some problems still exist. Sickle cell anemia is an example of what we call a point mutation. A point mutation means that there was just 1 point that was changed, right? Just one thing that got switched to something that we didn't want. 1 point, a point mutation. Sickle cell anemia is an example of a point mutation. Somehow, a different nitrogenous base was put in the place of the original one. Okay? We could also call this a substitution. So, something was substituted, right? We had one nitrogenous base in our normal healthy allele, and it was swapped. It was substituted for a different nitrogenous base. That's where the mutation came into place. Because it was just one point, we can call it a point mutation as well. So this type of mutation doesn't always affect the protein. You'll see sometimes we could have a mutation in the DNA, but it doesn't actually impact the protein because the amino acid stays the same. But in this case, it changed the amino acid, and so therefore it changed the protein. We're going to look at some other types of mutations. Okay, so not just sickle cell now. We're going to look at a frame shift mutation. Think about the term frame shift. Okay? This can make a lot of changes. So if we think about a long strand of a gene, a whole bunch of nucleotides in our DNA that we need to transcribe and then translate, they're all in codons, right? They're all in triplets in 3 pairs. Because when we go to translate our messenger RNA into a protein, it does it in codons, okay? Our triplet of messenger RNA, our codon, whether it's, let's say it's CAA, that's our codon. CAA is going to translate into one type of amino acid, but if I were to shift it over and do AAG, I'm going to have a completely different amino acid possibly. So if we take our frame and we shift it over, even just a letter or two, our codons all then change because we have to pair our triplets together in a different way at that point. So it's all about the codons. So frame shift mutations can be pretty catastrophic. We're going to look at a couple different kinds. We're going to look at deletions and additions. Okay? So first we're starting off with our normal DNA. There's no mutations. This is the healthy normal DNA. Your DNA codons are given to you, so CAA. Okay? What is your messenger RNA? Go ahead and transcribe that. And then you're going to translate it. What is the amino acid? Remember, you have to use your genetic code table for this. So you should be able to complete this table just like you did above, no problem. Okay? Then we're going to look at mutations. So our first one we're starting with is deletion. It says you're going to remove the very first nitrogenous base. So we started with CAA. Get rid of the C. Cross it off. That cytosine got deleted somehow. It got left out of the sequence. So you no longer have CAA. You're going to cross off the C. That means you're going to have to change your codons. So when you write your new DNA, your codons are going to be very different from what you wrote above. So the DNA sequence that I started with for a normal healthy adult was CAAGTAAAC. Okay? I delete the C. Then I have to rewrite my codons in this table. My new codons will now be AAG. That's one codon. TAA is my next codon. And my last codon only has AC. I need a triplet. I need a codon in order to get an amino acid. So if I only have 2, I'm not going to get an amino acid at all. Okay, keep that in mind as you go. So I now have my new triplets for my DNA, AAG, TAA. And my last one is incomplete, it's AC. Okay? Transcribe and translate. AAG turns into what for messenger RNA? That's your transcription. And then once you get that, look at your genetic code table and write down what the amino acid will be. Okay? Remember your last box, you're not going to be able to actually make anything. You're not going to be able to make your final amino acid because you only have 2 base pairs, not 3. That's an example of a deletion. We deleted cytosine. Okay? It shifted the entire frame of codons, which then changed pretty drastically the amino acids that we produced. We're going to do the addition mutation now. So your third table on this page is addition. So it says add a thymine to the end of your first triplet. So your first triplet now for your DNA should read CAT. So your DNA strand you had up above, you're just squeezing an extra thymine into that third spot. You keep all of your other letters. So you now have some new codons, okay? Your new codons for your DNA should be CAT. That's where we added it. Now we're keeping all the rest the same. So my second codon is now AGT. My third codon is going to be AAA. And then you might notice you have C left over, right? You have an extra guy on his own. Of course, one base pair is not going to code for an amino acid, so keep that in mind as you complete your table. You're now going to take your codons of your DNA, you're going to transcribe and translate. Write down what amino acids you're going to get, okay? Once you do that, think about it. Take a minute now that you've completed all three of your tables and look at the differences between each one. Why can addition and deletion frame shift mutations be such a big deal? Why can they be dangerous? If we have a normal healthy sequence of DNA and we accidentally delete a base pair or we add in a base pair, what's going to happen? Think about it. Take a look at the evidence that you now have, okay? You can probably imagine that if we have a long sequence of DNA that we're transcribing and translating, if the frame shift mutation happens very early in that sequence, it's going to shift everything after it, which could be catastrophic, right? We might not even end up with a protein at all. If the frame shift mutation happens at the very end of it, though, it might be okay, right? We might be okay. Just depends. So think about that for a minute. And then in the box, it says, explain why calling the addition and deletion frame shift mutations makes sense. Why do we use the term frame shift mutation? Why do we use the term addition and deletion? How does it all work? Give me a few sentences there. And to wrap up, it says this process, DNA going to RNA, going to a protein, is called the central dogma. Okay? DNA cannot leave the nucleus, it's too big. So that's why we transcribe DNA into messenger RNA. Of course, you all know messenger RNA is single-stranded. We're only coding one side of the DNA. We have our messenger RNA and it's much shorter. We're only transcribing a gene, just one section of the DNA that will make one protein, okay? That's why it's able to leave the nucleus. It's a lot smaller. That mRNA, messenger RNA, will leave the nucleus through the pores on the nuclear envelope, and it's going to go out to a ribosome. Ribosomes are where protein synthesis happens. So we use the messenger RNA template and we translate it, just like we're translating a language. We take the codons from the messenger RNA and we translate it into a new word, an amino acid word, right? We now have our chain of amino acids. This is our polypeptide chain, which is then going to fold and create a protein. Okay, we're now on part C. So part C, we're going to be talking a little bit about the skin. We're getting into some anatomy, right? So this, we want to think about the layers of the skin. If you didn't know, your skin actually has a lot of different layers. We talk about it a lot in terms of lacerations or cuts or burns. We want to know how deep the cut is, how many layers of skin did we cut through, how many layers of skin did we burn through, right? So knowing the layers is pretty important, especially in dermatology, especially in etiology on how we treat things. How deep we have to do our stitches or our sutures, how deep we have to put in different types of burn medications, skin layers, and such. So it says one of the most important functions of a cell is to make proteins. Proteins are sometimes kept in the cell, and sometimes they go out of the cell to work. The epidermis, part of your skin, is called a stratified squamous epithelium. It's a lot of words, okay? Stratified, meaning layers. Squamous, kind of a oblong shape. Epithelium. Our skin cells we call our epithelial cells, right? The only layer that undergoes cell division in that entire section of skin that we're looking at, the only layer that's actually dividing skin cells is actually the very bottom layer, the stratum basilae. Now, thinking about all those other layers, that's kind of hard to believe, right? If you guys remember, we were talking about through the cell cycle, through interphase, we talked about how the epidermis, the skin layers, are constantly replicating. They're constantly reproducing because we lose a lot of skin cells every day. They slough off, right? That's why we have dust in our house. It's all sorts of our skin cells that are just falling off. So that very bottom layer is incredibly important because they're the ones replenishing all of the cells up above. Okay? Now, as the cells migrate away from the bottom layer, as they move up to the surface of the skin, they start to die. But they leave the proteins behind, and the proteins, especially things like keratin, will form a protective barrier, a protective layer. And that's really what the skin does. It forms a protection for us. Certainly, yes, it has oil glands, it has sweat glands, it helps us regulate our body temperature and our moisture. But first and foremost, it acts as a barrier. So the layer on top, that stratum corneum, that's going to be mostly dead cells. And it's mostly just going to be the remaining proteins. They form a protective barrier. It's filled with keratin, lipids, fatty acids. Keratin is a fibrous protein found in our integumentary system. So an integumentary system is not only our skin, but also our hair and our nails. That entire system, we call it the integumentary system. It's a type of organ system. Okay? Keratin is incredibly important. Keratin is very high in concentration in your fingernails. You can think about how strong your nails are. It's a really tough protein. It also makes up your hair, right? Pretty tough. Keratin also makes up things like a rhinoceros's horn, right? So it can make these really strong structures. So having a little bit of it left in the outer layer of our skin is pretty important. The cells do die as they migrate to the surface of the skin, but the proteins and the fats remain as a protective mechanism. So answer these questions below. How many layers of the epidermis do you see in this picture? Name the layers from deep to superficial. So start with the deepest one and end with the most superficial layer. Double check your spelling. Number 3 says which layer is the only one that performs mitosis. So thinking about what you just read, which one does mitosis? And what type of tissue is the epidermis? This was also given to you up above. So what do we call the actual tissue? And then what organ system does the epidermis belong to? Well, we answered that as well. So write that down. Those are words you want to start knowing. Right? Now, last but not least, we have the analysis page. Okay, as we move through this class, we're starting to get you to write a little bit more scientifically on your lab reports. Okay, so instead of just answering a bunch of questions, we want you to put things together, to think critically about it, and to write a paragraph or two about what you found during your lab. So for this lab in particular, you're going to do a little bit of research. Research is an incredibly important stage of the scientific process, right? We need to know what's already been found out. We need to add to that data. We want to make sure we're using reputable, good sources of science, okay? So for this analysis section, you only have 2 sections that you need to answer, but I want paragraphs for each one. Give me at least 1 paragraph, complete sentences, double check your spelling, make sure you don't have typos, all right? You want to answer this in its entirety. So give me enough information to answer the questions fully, okay? Number 1, it says research sickle cell anemia. Discuss some of the health concerns related with this disease, okay? So don't tell me sickle cell anemia makes a sickle cell shape. Give me more than that. What does it actually look like for the people who have it? What does it feel like? What does it do to their life expectancy? What does it do to their bodies? Are there athletes with sickle cell anemia? Can you run? Can you climb upstairs okay? Can you have jobs? What does your life look like? Research a few things. Think about the health concerns. Does it make you more susceptible to other illnesses? I told you my friend's sister who has it, she's in the hospital a lot. Why might that be? Why would you have to end up going to a hospital for this disease? If you're not carrying enough oxygen, carbon dioxide through your blood, what would happen? So take some time to research this. Again, this is going to be on your computer. You want to look for .edu sites. You want to look for .org sites. Don't use anything like Wikipedia. You could also go to the Cincinnati State Library page and look up some peer-reviewed scientific articles and start looking at what the research is going on for this disease. So give me some different ideas. Discuss some of the health concerns. Okay, about a paragraph. Number 2, it says write out and try to answer a few questions that you have from this lab. Give me at least 3 questions. At least 3, okay? These are questions that you have. Try to answer them. So do some research and try to answer your questions. Write out what your question is and then write out the answer. If you cannot find it, if you ask a question that we don't know the answer to, write that. Tell me that. Say, this is what I'm wondering, but it doesn't seem like there's any research or data out there on it. Okay? Give me at least 3. This section here is to help you think critically and to be curious, right? A lot of science is all about thinking about what we know and what we still need to discover. So what are you curious about? Sometimes it takes practice to develop this part of your brain. We aren't taught to do this very often unless maybe you went to a Montessori type school. They're a little bit better at this. So for now, as scientists, we want to think critically. We want to be curious. As you go through healthcare and you take care of patients, you could use this type of practice to write case studies, to help your patients to think of ways that you can improve their experience in your care, and to do research around it, okay? So here are some ideas you could get started with. Maybe you want to research more about mutations. Maybe you have questions on the skin. Maybe you have more questions on sickle cell anemia that you didn't answer up above. Maybe you're curious about the central dogma, this whole transcription translation thing. Maybe you want to know more about different organ systems, okay? Whatever your questions are, research them, try to answer them. Make sure you give me at least 3 questions that you've written down, and try to answer those questions as best you can, or tell me if you couldn't find the answer. Okay, that's it for today. As always, reach out if you have questions. I hope you like this lab. I think it's a pretty interesting one. Have fun.